Hello friends, how are you? I think yesterday you must have enjoyed the respiratory system in different animals and in humans, and today we are going to go about the circulatory system. Yeah, I'm just having some seeds of some healthy food, and I'm wondering how this is going to reach to my cells. Well, I'm sure you are aware that whatever goes into my tube, digestive tube, from stomach and intestine, ultimately it's going to enter into blood, and from blood it's going to go into the different cells. So I think this can be a very good introduction to the topic of circulatory system in different animals and in humans, and lots of interesting things are ahead. Let's do with it. So here we are going to consider today a lot of interesting things about heart, blood vessels, lymph, blood groups and whatnot. So I can show you here now the circulatory system and circulation topic, today's topic, and we are going to consider details of the heart, human heart. I think through your school books from time to time you are well acquainted with the heart, human heart. We are going to say something about external features, internal structure and the working of the heart. External structure of the heart is heart morphology. Let's revise those things. Morphology is not only the external structure of the organism. Morphology can be external structure of any organ as well. The internal structure of heart, the section of the heart, you know, the two atria and the two ventricles and the valves, and that's all about heart anatomy. So we are going to understand heart morphology, heart anatomy, and then we want to know how does the heart work. You know how the heart works, contractions and relaxations, and it may be 70 to 75 times or even 80 times per minute, and that is heart physiology, how it works. So we are going to spend some time with the way the heart is structured and the way the heart functions. Of course, you are all aware that blood vessels are an important part of the circulatory system. You are aware of the arteries which carry oxygenated blood and the veins which carry deoxygenated blood. So we have a system of arteries and system of veins. Arterial system, venous system. These two, the arteries and the veins, they get connected to each other by capillaries. So arteries, they ultimately distribute the oxygenated blood to the cells, and then there the deoxygenated blood is collected, but at the tissue level, at the cell level, there are what are called as capillaries. So arteries and veins are connected by capillaries. So we are going to learn about heart, the blood vessels, and of course about the blood, because that is a fluid which flows through the heart and the vessels. And what are those things that we are supposed to know about blood? You are all aware the blood has two components. The liquid component of the blood is called as plasma, and the other things are cells, the blood cells. I am sure each one of you is aware of the red blood cells, RBCs, white blood cells, WBCs, and platelets. So these three types of cells make the solid component of the blood, and plasma will make the liquid component of the blood. So we want to say something about plasma and blood cells, and we can also imagine what are the different roles, different functions of the blood. Yes, many of you are curious to know about blood groups. Blood groups is one thing about which everybody is aware, but want to know a little more. Let us see how much of information about the blood group we can have. And yes, something about the lymph as well. So these are the things. In addition to these things, we want to compare the circulatory system in different animals. We will try to say what is an open circulatory system and what is a closed circulatory system. and then we will try to comment on the circulatory system as usual, because the three animals which we are going to study in details for the NEET later on, they are going to be earthworm, cockroach and frog. So we will have some information about the circulatory system of the earthworm, cockroach and frog. So we are going to consider human system as well as some information about these three animals. Please understand what is the purpose of our foundation course. It is just to be a little more aware. There are so many details. You don't need to remember everything, but you need to be aware of these things. I think this can be a good beginning to improve further your knowledge. You already are interested in biology, I know, but then that interest must be converted into the actual effect, and that is probably being done here. So these are the things we are going to learn. I think I would prefer to start with blood. Blood, how much of blood our human body has? Maybe 5 liters. Adult person will have 5 liters of blood. It can be less or more. And the 5 liters, how will you imagine? Probably you are aware of our milk pouches which are available. Usually the milk pouches are available in 500 ml, half a liter. You can imagine 10 such milk pouches. That's the amount of blood that we have in our body. It's continuously moving. And you are aware that this is so precious material. I can call this as a red gold. That will be red gold. Do you know black gold? Black gold you already know. What is that? The choroid. The choroid is something which is basically necessary for all our modern world, including transport, because all the petroleum and all we get from the crude oil. That is black gold. Then this is red gold. The regular gold, the metal that you already know, the shining thing. But then I think more than the shining gold, the metallic gold, this is red gold, which is important. It's very precious. It makes possible continuation of life. So we will now consider something about erythrocytes. Some more comments about these things I am going to make later on, but I will give you just a compressed information. Red blood cells, they are like this. They are kind of circular cells, but they are disc-like. And I think most of you are aware that these circular cells, they are like a flattened thing. You know, they are flat from both sides. You can imagine it's a circular cell which is flat from both sides. And one thing, at least in human case, it is without nucleus. Probably you know this. You may wonder, how can a cell survive without a nucleus? Well, interesting. In the beginning, when the cell is being formed, you know this very well, that red cells are being formed in the bone marrow, and when they are being formed in the bone marrow, they have a nucleus. And when they are about to be released into the blood, they are formed in the bone marrow and then they come into the blood. Before they enter into the bloodstream, the nucleus is removed. And that's how the red cells of the mammals, you know what mammals are. Only in case of mammals, the red blood cells are without nucleus. There is a good reason for this. Can you imagine why the nucleus is removed? Some of you are able to imagine. You know this very well, that the red blood cells contain hemoglobin. It's a special protein. It's called as a pigment protein, and it has got iron. Everybody knows that. So each red blood cell is packed with hemoglobin molecule. Millions of hemoglobin molecules. Imagine, millions of hemoglobin molecules present in every red blood cell. And red blood cell is very, very tiny. Even under microscope, it appears only as a very minute cell. And in that, millions of hemoglobin molecules are there. And the hemoglobin needs a space, because there are so many hemoglobin molecules. It needs a space, and that's why the nucleus is removed, so that more hemoglobin molecule can be adjusted. If you think of the red blood cells of other animals, fishes, frogs, lizards, birds, in these lower vertebrates, the red blood cells are with nucleus. But in mammals, because we are advanced than others, so naturally our red blood cells are without nucleus. That's a good thing. But then there are limitations. You know, it has a short life, about 100 days only. In your book, sometimes they mention 120 days and 80 days, and you may say, no, in our books it is 120 days. Some books it has 100 days, some books it is 80 days. Let me tell you, what are numbers you come into discussions in biology? Well, these are always plus minus. You don't expect that they are always exactly 100. No red cell will have exact 100. Some of the red cells may survive for more than 120 days. Some of them may get died before 100 days. It's simple. It's simple because red blood cells are continuously moving in the body, and because they are continuously moving in the body and they are transporting oxygen and carbon dioxide as well, it is obvious that they get exhausted and therefore they die. And it depends on the other conditions. So not exactly 100 days plus or minus. It's quite okay. We are all aware that oxygen carries. We are aware that hemoglobin carries oxygen. Fine. But please understand, the hemoglobin also carries carbon dioxide. So the red blood cells have double role: transport of oxygen as well as transport of carbon dioxide. So this is a little bit information about the red blood cells, and a lot of more information we can have later on as we will have this lesson learnt in our regular portion. Coming to the leukocytes, white blood cells, they are not white. They are colorless. They are wrongly called as white. You don't expect that they are white. They are not white. They are colorless cells, wrongly called as white blood cells. The real name is leukocytes. Let me break up this word. Leuko is colorless. Erythro is red. Leuko is colorless. Cytes means cells. Leukocyte is a correct term. They are colorless cells. And these leukocytes, they are definitely larger than red blood cells. Some of you may have probably understood the measuring of the cells in terms of microns. If you know this, it's good. 1 millimeter is divided into 1000 microns. So 1 millimeter is such a small distance. That 1 micron is divided into 1000 units, and then it is called as 1 millimeter. We are going to have some of these things in one of our later sessions, because we are going to have a session on cell biology. That time I think I'll make things more clear. So I want to say that the diameter of erythrocytes is suppose 8 microns. It's very, very small. And the diameter of the leukocytes, white blood cells, is say about 12 or 15 microns. So it is almost double the size. So WBCs are larger than RBCs. They are amoeboid in nature. What do you mean by amoeboid? Amoeboid means they move like an amoeba. These are circular. Erythrocytes are circular, disc-like. But then white blood cells are shapeless. They keep on changing their shape. And you are probably aware that WBCs can also go opposite in the direction of the blood flow. Suppose blood is moving from this point to this point, WBCs are an option. They can go in the same direction, or they can come in the reverse direction, or WBCs can even come out of the blood supply. They can come out of the blood capillaries and the blood vessels. Why? Because they have the major job of protection. So wherever the germs are there, they are free to go. So there are lots of interesting things about the WBCs which are very different from the RBCs. And just like RBCs, the WBCs are also being produced in the bone marrow. That process of production of the blood cells in the bone marrow is going on continuously, and that is being called as hemopoiesis. No. I'm sure you don't like terminology. I'm just deliberately trying to say biology learning is not about terminology. It's all about the concept. If you know that RBCs and WBCs are produced in the bone marrow, I think that is quite enough. But yes, as you go ahead, some terms you will have to pick up. What term I use? Hemopoiesis. If you like, good, you remember. If you don't like, forget. I will suggest 2 more terms. Erythropoiesis. Can you imagine? Yeah, erythropoiesis will be production of the red blood cells. And then can you think of a word which can say what is the what is white blood cell production. If this is erythropoiesis, this can be leukopoiesis. So erythropoiesis, RBC production, leukopoiesis, WBC production, and the two together, hemopoiesis. Oh my God, I don't think you will like to remember all these terms. If you like, you can write. But then if you write and remember, that's better. If you don't like these terms immediately, don't worry. I'll repeat only for the sake of your writing. Erythropoiesis. You want me to write somewhere? I can write somewhere. Okay, I'll write here. Erythropoiesis. Okay. Is this coming up in the record? Erythropoiesis, erythropoiesis. Similarly, you try to generate the word leukopoiesis and then the hemopoiesis. I think it's okay. If you know these terms, okay, otherwise you forget it. Anyway, I should not have gone into terminology, to be very frank. There are subtypes. You know, leukocytes or WBCs are subtypes. I'm sure in your school books you have learned this. And these subtypes are all well-known terms. Neutrophils, eosinophils, basophils. Shall I repeat? Neutrophils, eosinophils, and basophils. Similarly, monocytes and lymphocytes. I think you'll have to remember these terms now. Even if you don't know erythropoiesis and leukopoiesis and hemopoiesis, it doesn't matter. But you have to remember neutrophils, eosinophils, basophils, monocytes, lymphocytes. Overall, it seems that the total number of these cells is much lesser than the total number of red blood cells. Red blood cells are far more in number, but WBCs are comparatively less in number. And the neutrophils and eosinophils and basophils can be called as granulocytes. And monocytes and lymphocytes can be called as agranulocytes. What is the meaning? If there is a cell like a neutrophil or eosinophil, its cytoplasm will have granules. Granules are clusters. They are kind of solid materials which are scattered. Monocytes and lymphocytes, their cytoplasm is clear. They don't have granules. This is artificial classification. Each one of them has a role to play, and I think when you are going to start in details the study of the NEET and that type of biology, you are going to comment on the function of each. Just now I will simply say that one of the basic functions of these white blood cells is defense power. Neutrophils and monocytes are important. They have the ability to eat germs. So neutrophils and monocytes, they have the ability to eat germs. So wherever the germs are there, they will reach. Lymphocytes are very special. In fact, these are the most important of all the 5 types of cells, because lymphocytes can produce antibodies. You have heard of this word, antibodies. Antibodies are those which act against the foreign material, the germs. And our lymphocytes are all the time busy producing antibodies. And there are some lymphocytes which have the ability to produce some chemicals. And those chemicals also stimulate the entire system. So you are going to have a chapter on human diseases and human health. And in that case, in that chapter, we are going to learn something about immune system of the body. What do you mean by immune system? Immune system is that which is the defense system of the body. And the white blood cells, all of these play important role in the white blood cell, in the immune system, and probably lymphocytes have a major role. There are 2 types of lymphocytes. Again, I'm just mentioning. Don't worry if you don't know them, and if you don't want to remember, it's fine. But I'm just trying to mention there are 2 important types of lymphocytes. T lymphocyte. This is T lymphocyte, and then there will be B lymphocytes. Right, T and B. So lymphocytes may be T and B types, and they have their own role to play. The B lymphocytes are those which produce the antibodies. So these are the leukocytes, and the thrombocytes you have heard, and thrombocytes are also being called as platelets. They are very tiny, very small as compared to RBCs. They are smaller than even RBCs. And they have many other functions, but the major function is they help in their clotting. I think those details we can't go into just now. That's not necessary. So the main part of the blood therefore will be the cells, and these blood cells, they make 45% of the blood. If I just show you this way, suppose I draw here a tube. Suppose there's a tube full of blood. Then you can imagine, and if you centrifuge the blood, then the blood will be settled here, because when centrifugation will take place, the blood will be settling here, and there will be a colorless plasma which will be accommodated here. And you can imagine therefore this is going to be 45% and the plasma is going to be 55%. So this is plasma, because when you take blood and centrifuge it, if this is 100 ml of blood, then this will be 45 ml and this will be 55 ml. So this is the liquid part and this is the solid part. So we'll come to the liquid part. Plasma, 55% of the blood. It's colorless. And what is plasma? Plasma will have more of water. 90% of the plasma will be water. And that's how the blood is able to flow. Blood is the tissue that flows. It's written here. It is liquid connective tissue. You already know that the bone and the cartilage, have you heard of these things? Bone, cartilage, areolar tissue, adipose tissue. We're going to study these tissues a little bit later on. So bone, cartilage, areolar, adipose tissue, they are all connected tissues, but they can't move, they can't flow. You can't expect the bone to go from one place to another. You don't expect the adipose tissue, the fatty tissue, to go from one place to another. But blood is interesting. It is a connective tissue, fluid type, liquid type. It can move, it can go. So these are all the different cells of the connective tissue, and then plasma is the material which has got 90% of water, and it is this water that makes possible flow. So it has got special proteins. I'm just going to mention those proteins, and don't worry if you do not want to remember them. But some of you might have heard of some of the proteins in the blood. Have you heard of albumin, globulin? I'm repeating. Those of you, I think every time we have a session, you should sit with a notebook, because everything cannot be written on the board, and certain things you can listen and write as if you are in the classroom. And this is all a classroom itself, so you are getting almost the same direct experience. So I'll mention some of the blood proteins which you can record. Which are those special proteins? Albumin, globulin, prothrombin, and fibrinogen. I'll repeat. Albumin, globulin, prothrombin, fibrinogen. These are four important blood proteins, and they have some special functions. It is beyond the scope of the foundation course to go into details of these special proteins. You should have heard of their names. And there are lots of cations and anions. You know, a lot of electrolytes are there. So the plasma will have water, proteins, and cations, anions. I can name a few: iron, manganese, magnesium, calcium, sulfates, phosphates, chlorides, sodium, potassium, magnesium, calcium, iron. These are the cations. Anions, chlorides, sulfates, phosphates. These are the anions. And everything is present. You know, it's a very, very complex fluid. Plasma is very complex. Chemically, it's very, very complex. People have not yet been able to analyze the plasma constitution totally, because it will have all digested foods also. Plasma has all digested food. whatever the food I have just consumed in front of you in the beginning, I'm sure it's ultimately going into blood. The nutritive parts are going into blood, and which part of the blood is going to go into plasma. So all the foods that are digested, you have learned about the digestion, absorption. Absorption means, do you remember what is absorption? Absorption is entry of the digested food into blood. So blood has all the digested foods, which I have not written here. All amino acids and all the glucose and fatty acids, everything is there. Not only that, but all vitamins. When we consume food, all vitamins will be absorbed into blood. Not only that, but the blood will also have the oxygen and carbon dioxide because they are being transported. So all the major functions of the blood are being done by plasma. So to transport respiratory gases, to transport digested foods and hormones. My God, several hormones in the body. Think of the thyroxine. Plasma will have thyroxine. Plasma will have growth hormone. Plasma will have insulin. Plasma will have glucagon. When you say that blood carries the hormones, what do you mean? Which part of the blood is going to carry the hormone? Of course, it is the plasma. Cells can't do all these things. Cells have their own functions, but plasma has to do most of the other jobs. So that's about the plasma. Very complex fluid. I think I'll just comment on lymph. Lymph is something, in one sense it is similar to plasma. However, there are a few differences. Chemically it is rather slightly different, and it is something which flows through lymph capillaries. There are two types of capillaries exist in the body. We have blood capillaries and lymph capillaries. We are going to go to those details little later on. So presently I am just trying to make you aware that there is a fluid known as lymph, and just blood flows through blood capillaries, lymph flows through lymph capillaries, and lymph also has got great jobs. I think in one of the I'll take one of the opportunities later on to talk a little more about the lymph. I think this is what we have learnt about the blood, and there are a few more details about the blood, like blood groups and all, but I think that we are going to cover a little later today. So I think here we will have a small break. So let's now have something about heart. We will consider the heart of the topic, because heart is the heart of the topic, circulatory system. But before that, I want to put up a few things. I'm sure whenever you are listening to the lecture, number one, you must sit with pencil and paper. You must have actually a foundation, biology foundation notebook. Please make sure you must have this. These are all going to be very much useful to you, not only for your NEET, but also for all many more years of your biology career and interest. I think it was told to you that PDF, if possible, get them printed in case the facilities are available. There is a possibility of using 2 screens at a time, because if you are using the bigger screen, and you can use the TV screens also, because when we talked with some of the students and their parents, they say they are finding it all very interesting to look at the same things on the TV screen, and many people in the family can at a time watch all those things like dissections and all. Okay, so if you are using a TV screen, that is still better, and those kind of technical arrangements you should know, you might have known, and that you can. Otherwise, a laptop screen. A tablet is okay, but then I think better than a smaller screen. Otherwise, if you don't have this facility, then you have to use your regular mobile screens, and that is quite okay. If you have 2, then what you can do is, suppose on one screen you are observing the things enlarged, and on the other screen, if you have some PDF, you are reading side by side, that's possible. Don't worry much about all these things, as long as you are able to concentrate. Even the small mobile phone is also good. This is one interesting thing, and some of you may be knowing and some of you may not be knowing. Whenever you are observing the lecture being loaded on the YouTube, there is a possibility, if you choose correctly the option, you will find that captions are available. And many of you, of course, must be looking at the recorded lecture with captions. Those captions are auto-recorded. And sometimes, if I use some Marathi terms, then probably those captions, they get jumbled. That's quite okay. But otherwise, the English captions are there. So, these are the things some of you know and some of you may not be knowing. So, I'm just giving you these points. Yeah, the worksheets are important. I think we have sent you worksheets for 4 sessions now. No, today is 4th. So 3 sessions at least we have sent. And usually we are sending you 2 worksheets for every session. 2 worksheets for digestion, nutrition, and 2 worksheets that we have sent for respiration just today, some time back. And this recording is going on. So we will be able to release this recording tomorrow morning. I think that instruction is also already sent to you. So I mean to say that you should be solving those worksheets. They are very important. Many of you may be thinking, how is it that you will be able to contact us for your difficulties? Well, let me be very frank, there are some limitations here. But then, if you read the message carefully that was sent yesterday evening, then you will find that we have suggested you to send your so-called specific doubts on the Google Classroom platform. You can just type right, okay, and just send those things to us. We will try to answer to those doubts. And then we are thinking that we will also arrange a special session only for doubt solving, but even then it is not going to be not like the Google Meet, or it's not going to be Zoom, because there are lots of difficulties there. So what we will do, we will collect all the doubt that you are posting to us, and then we will be using those doubts to explain the thing, just as now I am explaining to you the whole thing. We will record all explanations of your doubts. I think that will sound good. Many of the parents and students of the foundation course are reporting that they are very happy. There is a continuity of the contents, and the sound and the light quality are good. I mean to say, there are many ways it is possible to conduct online sessions. But we have thought carefully about this, and we have deliberately avoided Zoom and Google Meet, and it seems that this is all good for you. Maybe that the contact between us is not possible immediately, but it can be made possible through the questions that you are posting to us, and we will hold sessions and something can be done. Feel happy? Nice. Let's now go for the details of the heart. So here we will have some details about the heart. You can understand the position of the heart in the thoracic cavity. You have thoracic cavity and you have abdominal cavity. So in the thoracic cavity, the heart is in the center, as you can see here, and the tip of the heart is towards left side. Many times it is wrongly said that the heart is on left side. No, heart is never on left side. The heart is in the center, but its tip is directed to the left side, and it is between the 2 lungs. And there is a space nature has generated for heart specially. So what is this thoracic cavity? Thoracic cavity is bounded by ribs, and there is a rib cage, you can say. Down below there is diaphragm, and behind there is vertebral column. Vertebral column behind the rib cage here, and the rib cage is all connected to sternum here, and the diaphragm below. It's a perfectly safe place in which these 2 lungs and heart are lying. So that's peculiar. Let's go for the structures of the heart, and we can have external and internal structures. So we will have first the external view, that is the morphology of the heart. Look at this morphology of the heart. This is dorsal view. You can look at the heart dorsally and ventrally. Yes. We are going to conduct a small practical sessions for this. We have heart, of course not human heart, but goat heart. You have seen the practical about the goat respiratory system, and now we have a goat heart, and we'll show you the details of the goat heart maybe in a day or 2. That time you can understand what is dorsal and ventral view. When you say dorsal, the back view. So if you are looking at my heart from the back side, it is dorsal view. And the ventral view, if you are looking at the heart from my my heart from your front side, from my front side, that is the ventral view. So this is our ventral side and this is our dorsal side. So actually, more technically, this is better called as anterior side and this is posterior side. But I think those details will be out of place just now. Let us simply say this is the ventral side. So ventral view and dorsal view. This is the dorsal view, and you know the heart is having chambers, atria and ventricles, and this is a right atrium, and this is going to be the left atrium on that side, and there are two ventricles, right and left ventricles, and these are all several blood vessels which are attached to the heart. I think what you should know definitely is the aorta, the dorsal aorta, which can also be called as aortic arch, and there are some veins, superior vena cava, and there is an inferior vena cava. I think those details can be learned later on. But interestingly, there is a coronary circulation. You can see here coronary sinus, which is actually draining the blood from the different parts of the heart. I think you need to look at a better diagram. I know you will be very happy to see this one. This is good. Same thing, but colors make a different sense to us. Same thing here. So you can see this is the coronary sinus. You can see the coronary circulation. You know, coronary circulation is that which is about the blood supply to heart. You're all aware that sometimes the blood supply to heart may not be good in some people. There may be problems. So it is all about these. You can see the red things. They're all coronary circulation. Arteries, blood is being supplied by coronary supply. I mean the artery supply, and blood is all collected by the veins. So these are the blood vessels. You have the dorsal aorta, the superior vena cava, which is collecting blood from the superior side, that is upper side, and this is the inferior vena cava, which is collecting the blood from the lower side. More details we can learn. I think we have studied two dorsal views. Now this is the ventral view. This is from the front side. If you look at the heart from the front side, yeah, these are the two chambers of the ventricle, superficially seen outside, the left ventricle and the right ventricle, and you have the right auricle and left auricle. Auricle, okay, sometimes you can call it the auricle, but better term is atrium. So right atrium and left atrium. And now this is the major blood vessel which is making a curve, which is turning backside, and that is called an aortic arch, the main artery of the body which supplies blood to different organs. Then this is the superior vena cava, collecting blood from superior side, inferior vena cava collecting blood from inferior side, and this is then the coronary artery, and you know coronary arteries supply oxygenated blood to the heart. Some of you must be feeling dizzy. You may be wondering, is it all this that you want to remember? Please, you don't have to remember everything. You should have heard. You know, this course is meant for bridging the gap between 8th, 9th, 10th and 11th, 12th. Some of you might have learned this in some details and some of you may not have learned it at all. It really doesn't matter. We all have equal opportunity to learn it now. So this is the ventral view. I think I'll comment a few things and then I'll proceed again. I'll comment it like this. That will provide a good opportunity for this machine to get cooled. And you know what is this machine? Can we show this machine to them?
Sure, just a minute.
So here is you know, these transparencies. This is called as a transparency, and the transparencies are put on the machine. Maybe that you already know this machine. This is called as overhead projector, OHP, and this transparency can be put here like this, and there is a bulb, and there is a lens here, and it gets projected. Yes, we have a LCD projector, the more commonly being used, and you have the OHP machine also. But you know, the only problem is that it gets heated if it is used for a longer time. But then we thought that it is better that you should know what are the things that are being used. Okay, let's begin. I'll put off this live. This is L1. I'm putting it off, and now I'm starting once again. Yeah, that's it. So it's a very nice way to learn. In the regular class, you know, when there are 100 students and even more than 100 students, not everybody is able to observe clearly. Now, you are having that opportunity during this foundation course to have these things seen directly. And to many of the parents whom we are trying to contact or they are contacting us, they are so happy that they say, please continue like this throughout the year. Oh my God, that will be a great responsibility, because recording these things takes a lot of time. But if you want all that to happen that way, we will be really very much happy about that also. So we have now internal structure of the heart. The heart has been sectioned. The heart has been sectioned, vertically sectioned. The two atria and the two ventricles. Right atrium, left atrium. Right ventricle, left ventricle. Right atrium, left atrium. Right ventricle, left ventricle. Four-chambered heart. The two atria are separated. That separation, septum is not seen, and the two ventricles are separated. You can see the septum here. What do you mean by septum? It's a partition. It's a muscular wall. There is a muscular wall between two ventricles, and similar muscular wall is between the two atria. Why that is not seen? Because this blood vessel is covering that partition. So if I remove this blood vessel, then you can see the continuity of that partition. So the atria, the two atria are separated from each other, and the two ventricles are also separated from each other. There are several levels. And I know that you have gone through the different boards, and some of the board books have this kind of a diagram, and some of the board books that you have used may not have this diagram, and it is quite possible that your teachers have taken trouble to go into more details. It is also possible that while you were in 10th, you have used some extra material, extra books, etc. So you should generally know this. What else I must say? This is a muscular thing. Atria are thin walled. See, this is an atrium. Each atrium is thin walled, and ventricle is quite thick walled. Can you say why ventricles are thicker? Just imagine. I think some of you want to say that ventricles are thicker because they have to send the blood to long distances. Atria, when they contract, the blood will be sent to ventricle. So it's only a few centimeter, and so the pressure that the atria must generate need not be more. So with lesser pressure, atria can send the blood into ventricles. But when ventricles contract, they have to send the blood to long distances. Let us think what happens to the blood which is in the right ventricle. Just think. Right ventricle, when the two ventricles of course contract together, blood from right ventricle will enter into what is called as pulmonary aorta, and from there it will go into two branches. These are all going to the two branches are connected to the lungs. These are all being called as pulmonary arteries. Once again I will say, blood from the right ventricle will enter into pulmonary aorta, and from pulmonary aorta it will go to the left lung and into the right lung. Why? The blood which is here is all deoxygenated, but it has to send to the lungs. Lungs are okay, directly nearer, but not. The distance is there, some distance is there. It's not as easy. So atria have a very easy way to push the blood into the ventricle. But this left ventricle has to send the blood through these pipes to the two lungs. And when you think of the left ventricle, my God, imagine what kind of blood is there in the left ventricle. You have learned some of these things. The left ventricle blood is all oxygenated, and this oxygenated blood will enter into this pipe. This pipe is called as dorsal aorta, and it is a major artery, and that will give blood to entire body, almost entire body. It means that this ventricle must be more thicker. Yeah, it is true. Can you carefully observe? The right ventricle is thick and the left is also thick, but the left ventricle is more thick than the right ventricle. Obviously, because the blood has to be sent from here to all parts of the body. So that's how things happen. What more you must say about heart? I think I can show you a better picture about the heart, and that will also provide the machine to take a rest for a few minutes or seconds. I'll show you a better picture now. and we will try to revise these things. I think understanding about human heart is so good, so important, and you otherwise also are aware that two organs in the body are extremely important. One is heart and another is brain. Okay, they have their own specialities, and don't forget that many of you used to say, or many of you may be saying even today, that you want to become cardiologists. This may be a good beginning for you to become a cardiologist. Let's have a slightly different diagram. Again, similar things. There are some. Let's see how the circulation will be taking place. This is the right atrium, and in the right atrium, two pipelines are opening. This is one pipe. I'm saying it pipe because it's really pipe-like. It is a tube. Superior vena cava. Blood from all the upper part of the body is collected by superior vena cava. You can call it as precaval. Sometimes in books they give it precaval, but better term is superior vena cava. And the blood enters here. Follow the arrows. And there will be blood coming from the lower side of the body. Blood from all the lower side of the body will be coming again here, and this is called as inferior vena cava. You know, this is a term. Superior part and inferior part. So, superior, inferior. So, all the blood from inferior parts of the body is collected by inferior vena cava, and these two veins, major veins, are opening into right atrium. And blood from the right atrium will enter right ventricle. Follow the arrow. But then, of course, you know that there is a valve here, and it is called as tricuspid valve. I think you are aware that between the atria and ventricle there are valves, and these valves are membranous, and they have threads which are being called as chordae tendineae. I know many of you do not want to listen to these terms. Some of you are likely to develop a negative approach to biology because of terms. If you don't want to remember the chordae tendineae, simply say that the flaps are there, and then there are thread-like structures are there. You know, what is the purpose of all this arrangement? Blood should enter from atrium to ventricle, but it shouldn't go back, because you want that the blood which has entered from atrium to ventricle must go into the pulmonary aorta, pulmonary arch. So that's how these things work. So there is a tricuspid valve. It is called as tricuspid because it has got three flaps. Similarly, blood which is there in the left atrium, it is collecting blood from the lungs. I'll repeat. Blood which is deoxygenated, which has come from all body parts into the right ventricle, will enter into the pulmonary aorta. This pulmonary aorta will have two branches going to the two lungs. And you know, yesterday we have learned, when blood goes into the lungs, what happens? What is there in the lungs? Finally, the lungs have balloon-like small things known as alveoli. So blood goes there, and blood gets oxidized. So the RBCs will pick up the oxygen, and the oxygenated blood will then come back to the heart. Once again, the two pulmonary arteries will be going to the lungs. This is something that most of the students know, that pulmonary arteries contain deoxygenated blood. Although they are arteries, they are collecting, they are carrying deoxygenated blood. Why? They are taking the blood to the lungs for oxygenation. Rather than purification, let us use the word oxygenation. So the oxygenation occurs on the surface of the alveoli. The hemoglobin catches a lot of oxygen, and all that oxygenated blood is coming to the left atrium. While going, the blood is going through one tube. Each lung only one tube, known as the pulmonary artery. But while coming back from the lungs, it is coming by two blood vessels. Two from the right lung and two from left lung. Now it is coming to the heart. So the blood is coming to the heart, and you generally are aware that the vessels which come to the heart are called as veins. So pulmonary veins, total 4 in number, and they are opening into the left atrium. So although these are veins, the blood is oxygenated, and although these are arteries, the blood is deoxygenated. Yeah, this is something many of the students remember, I don't know, this is also a little complicated, but they do remember, and if you don't, if you have not remembered till now, please try to understand it. So pulmonary arteries carry deoxygenated blood, and pulmonary veins carry oxygenated blood to the left atrium. From left atrium, follow the arrow, blood will enter into left ventricle, and this gateway is closed and opened as per requirement by a valve which is called as bicuspid valve. So there is a tricuspid valve here and there is a bicuspid valve here. Fine. And when the ventricles will contract, when both ventricles will contract, blood from the right ventricle will go to the lungs and blood from the left ventricle will go to the entire body. We are going to learn a little bit about this, that when contractions occur, first atria contract. Probably some of you know that when both the atria will contract together, it is called as atrial systole. Systole, what is that term? Systole is contraction. After that, the ventricles contract. What do you call this as? Ventricular systole. So atria contract and then ventricles contract. So the heart contracts. And when the atria relax, after some time even the ventricles relax. And we are going to talk something about contraction and relaxation later on. So those are the events of the cardiac cycle. What is a cardiac cycle? It is something which happens rhythmically and happens in a very regular manner in a healthy heart. And we'll talk about that, a little bit about that later on. So just revise these things. Let us have opportunity for this machine to take rest for a while, and then same thing you are going to observe in the colored picture. So can you imagine, and I'm just going to repeat a few points for you. Can you say what are the four chambers of the heart? Two atria superior, two ventricles inferior. Atria are thin walled, ventricles are thick walled. Oh my God, some of you have already made two columns and you have started writing difference between atria and ventricles. Okay, that's a good idea. Okay, let us make two columns. Yeah, make two columns. One side atria, one side ventricles. Fine. I can see some of you doing it. You may say, how can I see that? I can imagine. So, atria, ventricle. Atria thin walled, ventricles thick walled. Atria upper, that means superior chambers, ventricles inferior, lower chambers. Atria are collecting chambers because blood is being collected in the two atria, and ventricles are distributing chambers. I repeat the points. Atria upper, that is superior chambers, ventricles inferior chambers. Atria as compared to ventricles, they are thin walled, ventricles are thick walled. Atria are collecting chambers, ventricles are distributing chambers. I think some such writing you can do, you can imagine, and some of you might have thought of comparisons are good because comparisons make possible better understanding of the same topic. So we will have now, once again we'll have the same picture. I think you may like it a little better than the earlier one. Probably it's a good way to revise, because heart is something about which I think all of you should know, because your age is such that you become an easy victim of hearts. Well, that's a joke only. Don't take it seriously. So here is the heart, and you have the two atria and the two ventricles. Ventricles from inside is having the rough surface, and you have this tricuspid valve and the bicuspid valve, and you have the threads known as chordae tendineae, and you have from the right ventricle, this is a pathway, this is the pulmonary arch, pulmonary aorta you can say, going to the two lungs. Blood from the lungs is being collected on each side by two pulmonary veins, and all those four pulmonary veins are opening here, and from the left atrium the blood is coming into the left atrium, and so on. So this is once again a kind of a revision. The pulmonary aorta, this one, the yellow one, and aortic arch, the red one, which is passing from behind here. At the start, they have what are called as semilunar valves. As you can see, semilunar valve. Semi means half moon, semilunar. As you can see, it's half moon here. Actually, they are like a they are like a half sphere, but then in this section they will appear to be half moon shaped and so on. So that's what we have tried to understand about the heart, and this is the last picture about the heart to help you to understand the valves. Suppose you are taking a section, suppose the heart is being sectioned like this, and then you can see the tricuspid valve and the bicuspid valve. Probably you already know that bicuspid valve is also called as mitral valve. So tricuspid valve, bicuspid valve, and these are the semilunar valves which are at the beginning of the two aortic. And this is not very important for you, but this is the surface view of the various valves. I think we'll take a small pause here. So here we are to understand the working of the heart, physiology. Morphology I learned, anatomy I learned, now working. When you think of working of heart, working of liver, working of stomach, working of an eye, that working is involving some mechanism, okay? And that study of the working or the mechanism of working is what is called as physiology. So let's have a little bit understanding about the physiology of the heart. The heart has one peculiarity, and probably you have heard of that. And that peculiarity is that the heart has its own brain. My God, what am I saying? I mean to say, heart doesn't depend on the brain to send signals for beating. Are you understanding what I am trying to say? Heart itself has got a tissue, specialized muscle tissue in the heart, which acts as a pacemaker. Have you heard of this word? If not, don't worry. There is a tissue in the wall of the right atrium, in the dorsal wall of the right atrium, which has got the ability to send signals. Signals? What signals? Of course, electrical signals. And for what purpose? To bring about contraction. This is very important mechanism, because if heart beats only after getting signal from brain, it will be havoc, because it is possible that in the day-to-day working, brain may forget to send a signal to the heart. Forget, I mean to say, there can be problems of signals being conducted from brain to heart for each beating of the heart. So nature has designed this very great mechanism right from fishes to mammals. That means entire vertebrate group, the heart is said to be myogenic. This is something that you can record. M-Y-O-G-E-N-I-C. M-Y-O-G-E-N-I-C. Myogenic means what? What is the meaning of the word myogenic? Myogenic means the heart has the capacity to generate signals by itself. So it will produce a signal at a regular frequency. A heart will produce those signals, say ultimately 70 times a minute, 72, 75. Some people can have a faster heartbeat. They have 80 times a minute signal produced, and those signals, they are transported. So in the heart tissue that you have learned, the signals are transported because the signals are beginning from atria. The atria will contract first, and then the signals will go into the ventricle, and the ventricles will contract after atrial contraction. I'll repeat what I am trying to say. This particular spatial tissue which has the ability to generate the signals, it is present in the right atrium. So from there the signal begins and it will spread on the two atria, and after the atria the signals will go into the ventricles. So as the signals are first received by the atrial muscles, the atria will contract, and later on the ventricles will contract. I think I had introduced these terms to you: systole, diastole. Let us have a look at this particular picture. This is ventricular systole. What do you mean by systole? Contraction. And this is ventricular diastole. What does it mean? The ventricles are expanding. It's very interesting to observe here that when ventricles are contracting, the atria are already relaxed, and when the atria are contracting, ventricles are relaxed. It is not happening exactly like this because there is something which is called as common diastole, about which I am not going to say anything just now. But overall, as a beginning part of the understanding of the working of the heart, you may generalize a statement like this, that atricular relaxation, that time ventricular contraction. Atrial contraction, that time ventricular relaxation. One after another. So atria contract, then the ventricles contract, and so on. This is good because when atria will contract, the blood will go into ventricles. And when the ventricles will contract, then the blood will go into the pulmonary arteries, will carry it to the lungs, and the aorta will carry it to the entire body. So this is first, and then this is the second thing. So that's how systole and diastole. What is the meaning of systole? Systole is contraction. And what is diastole? Diastole is relaxation. Sample wise, here it is ventricular contraction, so ventricular systole, ventricular diastole, that is expansion of the ventricles. This is very nicely regulated. It is all regulated by the system, I told you, myogenic system. Of course, brain is also sending signals to heart, and brain can modify the working of the heart additionally, secondarily. This all leads to what is known as cardiac cycle. What is cardiac cycle? Same thing, sequence of events that are taking place in the heart. What are the events? Contraction of the atria, relaxation of the atria, contraction of the ventricles, relaxation of the ventricles. Major four things. I'll repeat: contraction of atria, relaxation of atria, contraction of ventricles, relaxation of ventricle. So, ventricular systole, diastole, and atrial systole and diastole. There are definite time intervals available. Say for example, if my heart is beating say 70 times in 60 seconds, 70 times in 60 seconds, 1 minute, then what will be the duration for one heartbeat? It will be obviously 60 upon 70. 60 upon 70 seconds. What does it mean? 0.8 seconds. I'll repeat my statement. In 60 seconds, suppose my heart beats 70 times, it means every heartbeat will be completed just in 0.8 seconds because it is 60 upon 70. And can you imagine, all these four events are completed in just 0.8 seconds. Fantastic. Of course, suppose you are having exercise, or suppose you are trapped in a situation when you get frightened and heartbeats increase, then of course the heartbeat will be completed in a shorter time, and so on and so forth. So this is the concept of systole and diastole. Actually, the systole and diastole of this kind will lead to what is called as double circulation, and I'll just let you know firstly what is double circulation. Here the double circulation will mean that heart will have blood passing through it 2 times in one course of circulation cycle. I think I have to repeat this statement. I am talking of double circulation. Basically, the human heart, the mammalian heart, and also the heart of the birds has 4 chambers very clearly. You already know that amphibia, frog, it has only 3-chambered heart, but birds and mammals have 4-chambered heart, and the right side of the heart is totally separated from the left side. Now what happens is that 2 circuits of circulation are generated here. Imagine, one circuit is from heart to lungs and back from lungs to heart. That's called as pulmonary circuit. I'll repeat, heart to lungs, lungs to heart. Heart to lungs, you know this very well, the pulmonary arteries which are carrying deoxygenated blood. Now from lungs back to heart, You know this very well, that from each lung there are 2 pulmonary veins, and the total 4 pulmonary veins join the left atrium. So this is the kind of circulation that goes on between heart and lungs, and that is being called as pulmonary circuit or pulmonary part of the double circulation. I tried to explain to you the pulmonary circulation just now. Let's have pulmonary circulation once again revised. Blood will flow from right atrium to right ventricle, and from right ventricle it will go to the lungs via the pulmonary arteries, and the blood will get oxygenated, and the oxygenated blood will enter into the 4 pulmonary veins, and this represents 4 pulmonary veins, and it will go into the left auricle, better called as left ventricle. So this is how pulmonary circuit is between right ventricle and left auricle or left atrium. So that is called as pulmonary circuit. Now let us understand which is the other, that is systemic circuit. This is pulmonary circulation and this is the systemic circulation. Systemic circuit consists of the flow of blood from left ventricle. The blood will start. Arteries, the dorsal aorta, the major aorta will take the blood from the left ventricle and it will carry it to all body parts. After a while we are going to see the arterial system. That time you can understand this better. So all the oxygenated blood which has already been received from the lungs now goes to all body parts. In almost every body part the blood will go, and the oxygen will be used, and then the carbon dioxide will be produced, and the deoxygenated blood will start traveling back to the right atrium. You know that there is one superior vena cava and one inferior vena cava. So through the superior and inferior vena cava, the blood will be collected from different body parts and it will enter into right atrium, that is right ventricle. And then you know already from right atrium it will go to the right ventricle and then it will go to the lungs. So what is systemic circulation? Systemic circulation is between left ventricle and right atrium. This circuit, that is called as systemic circulation. And this is what is called as double circulation. Why it is called as double circulation? In one course of cycle of the blood movement, blood is once traveling through the right side and then it is traveling through the left side. In one complete circulation, suppose the blood starts from right auricle to right ventricle to lungs, lungs to the left atrium to left ventricle to the body parts, and body parts back to right atrium. Very clearly, the blood has travelled twice through the heart. Once it has travelled, suppose you are sitting on the red blood cell, imagine. Suppose you are so small that you are sitting on the red blood cell and you are left in the right atrium, and suppose you are asked to come back to right atrium. Theoretically, you can imagine that first you will have to travel between the right side and the lung, and then you will travel between the left side and the body part. This is possible because the complete partition is there. So the right side is totally separated from left side, and therefore typical double circulation, I can show you another better picture of the double circulation. Typical double circulation of this kind can be expected to take place only in that particular design of the heart where there are clearly the right side separated from the left side. In the case of amphibia, which has got 3-chambered heart, you are not expecting this to happen, because you are expecting that there are 2 atria and 1 ventricle. So naturally you will expect that the blood is getting mixed in the ventricle. So double circulation is not possible in amphibia. In the reptilia, in some reptiles, heart by chance can be 4-chambered, like crocodiles and turtles, they have 4-chambered heart. But most of the other reptiles have 3-chambered heart, slightly 3.5 sometimes it is said. So I think serious double circulation is possible in birds and mammals. So that's double circulation. There are advantages of double circulation. One major advantage of double circulation is because the oxygenated and deoxygenated blood is not mixed, I think therefore all the body parts are supplied with the best type of blood, in the sense the blood that has got enough oxygen. In the case of the amphibia, I am going to show you the picture of the amphibian heart. There are 2 atria and 1 ventricle, and the blood gets mixed. Naturally, there is lesser oxygen supplied to the tissues of the frog. Anyway, so I hope you have understood double circulation. This is circulation between heart and lungs. This is pulmonary circuit, and this is the circulation between heart and body parts. This is pulmonary circulation because lungs are mentioned here, and this is the circulation between heart and body parts. The head is also included. The rest of the body, lower side of the body is also included. So all this is systemic, because system is between all body parts and heart, and pulmonary is between heart and lungs. So there are 2 things you can imagine. I mean to say, you can think of 2 things while explaining the physiology of the heart. One can be the cardiac cycle. When I say cardiac cycle, I am trying to say the systole diastole cycle. And second thing is double circulation. You can consider that double circulation is also a part of the working of the heart, because the blood has to flow through the heart 2 times. A lot of evolutionary changes have occurred, and I think I can show you a picture where the heart chambers have undergone variation. The simple heart of the fish has 2 chambers, and the complex heart of the birds and the mammals have 4 chambers. That's one thing that I can show you just now, and later on we can go to the arterial and the venous system. Here I take a small pause. This is what I was trying to say. The heart has undergone a lot of evolution. When fishes gave rise to amphibia, and amphibia to reptilia, and reptilia, from reptilia 2 groups have been formed, and that is birds and mammals. Most of the things have undergone change. Heart has undergone a very beautiful change. Let us have a look at this picture, and this is about comparative anatomy of vertebrate hearts. What do you mean by comparative anatomy? Simple. Internal structure studied in comparison. Here is the fish heart, the frog's heart, a reptilian heart, and the bird heart, and mammalian heart. You know, the 5 groups of vertebrates. Interestingly, fish has only 2 chambers. So fish has 1 atrium and 1 ventricle, 2-chambered heart, and it has got only deoxygenated blood. From the fish heart, the blood will go directly to the gills. So blood will come into the atrium. From atrium it will go to ventricle, and from ventricle it will go to gills, and from gills it will go directly to all parts of the body. So venous heart, it is called as venous heart because fish heart does not have any arterial blood. Only venous blood it has got, and the venous blood will go to the gills for oxygenation. From gills it doesn't come back to the heart. From gills it will go directly to body parts. So oxygenated blood will go to the body parts. There is one additional chamber, however, to help this process. This is known as sinus venosus. If you remember, good. If not, you can think this is what is called as sinus venosus. All the blood from the fish body will be coming into sinus venosus. From sinus venosus it will go to atrium. From atrium to ventricle. And from ventricle to gills. So 2-chambered heart. During evolution, when fishes, some of the ancient groups of fishes were transformed into amphibia, the 2-chambered heart became 3-chambered. You know this fact very well, that frog's heart, amphibian heart, has 2 atria and only 1 ventricle. So you can imagine blood from right atrium, which is expected to be deoxygenated, will enter here, and blood from left atrium, which is oxygenated, will enter here, and all the blood will get mixed. And see, 3-chambered heart, the blood gets mixed, and from here the blood will be provided to all body parts, and it's going to be mixed blood. Okay, amphibians are happy, they have no problem. Their body tissues are adapted for that kind of mixed blood, but evolution wise it is still a lower group. From amphibia, reptiles were evolved. Reptiles, what do you mean by reptiles? Lizards, snakes, turtles, tortoises, crocodiles, and those ancient dinosaurs. They are all reptiles. Today's reptiles, many of them have two atria and partially divided ventricle. Compare this with this. This ventricle is totally undivided, whereas here the ventricle is partly divided. But it's of no use, because anyway blood is going to get mixed. So there is no special advantage, but this is one stage in evolution. So people say this is 2-chambered, this is 3-chambered, and reptilian heart sometimes they say it is 3.5-chambered. What do you mean by 3.5? 3.5 because the ventricle is partially divided. It's a stage in evolution. Some reptiles like crocodiles, turtles have this particular partition complete. So some of the reptiles like crocodiles, they have 4-chambered heart. So you can expect that they are having a better oxygen supply to the tissues. Coming to the birds and mammals, very clearly. Birds and mammals have clear-cut 4 chambers, which we have already considered. So that is how the heart has undergone evolution. It is not only the heart. Almost every organ system has undergone change, that is evolution, during the course of millions of years. And let me tell you that it is very, very necessary to have a constant thinking about evolution while anything in biology is being studied. How and why we will come to know as we go ahead. So it is the evolution that provides the necessary logic for understanding biology. So with this, I think we can consider now a little more details about the frog heart, because I think we have told you repeatedly there are 3 types of animals, or you can say 3 animal types you are going to have for your meat, that is earthworm, cockroach and frog. This is typically the frog's heart, and 3-chambered we have just now considered. The right atrium, the left atrium, and this is the ventricle. These are not, these are all muscles, muscle blocks which are projecting, but the ventricle is not divided. That's it. Now lots of complexities are there, and you are not to be worried about it. Some details about the frog's heart you are going to learn at a proper time in the regular course. But you should know this. That's all. And this is the sinus venosus. Just like fish has sinus venosus, even the frog's heart has sinus venosus. So blood from frog's body will be collected into the sinus venosus. From there it will go into the right atrium, etc. Additional chamber is there on the ventral side, which is called as truncus arteriosus, this flask-like thing. So frog's heart is much primitive than our heart, but it has got some extra chambers, and you don't have to worry much about this. So frog's heart, and the blood is getting mixed, and the mixed blood is going to get into the entire body of the frog. So I think we will now be in a position to take into account now the arterial and the venous system. So we have already considered blood, then we have considered the external structure and internal structure of the heart. Then we have considered the working of the heart in terms of systole, diastole, and also we have commented something about the double circulation. Not only that, but we have taken into account the evolution of the heart, how the design has become simple to complex. So blood and heart, I think you have some good revision idea. It is now time for us to take into account the blood vessels. And when I talk of blood vessels, I am trying to say the arteries and the veins. This is being given the name circulatory system because everything is here. The heart is there and the blood vessels are there, and you can imagine even the blood is there. That's how this picture is being given the name circulatory system. But if you want to analyze some details, we have already considered heart and the blood. Now consider the tubes. All the red colored tubes which are shown are obviously the arteries which are carrying oxygenated blood. So we have already studied some of the details of the blood, and then we have tried to consider the details of the heart, the external structure, the internal structure, and the physiology, the working in terms of cardiac cycle, and also related thing, that is double circulation. Not only that, but we have also taken into account the way the heart has evolved from simple heart of a fish to a complex heart of the mammal. I think now it is the right time to take into account the blood vessels. This picture is being given the name circulatory system because in this picture you will observe the heart and all the blood vessels. Of course, you assume the blood is there. We will not talk about heart anymore directly. We will now start talking about the red and the blue pipes which are shown here. These pipes are blood vessels. Red pipes which are starting from heart and distributed in the entire body. These are the arteries, that's arterial system. And all these blue blood vessels which are collecting blood from the different parts of the body and bringing it to the heart, those are veins, and they are making its own system, venous system. So in this picture, this combination of arterial system and venous system. I think let's go for details, some details of the arteries, the arterial system. Here is the arterial system. There are so many names. I don't think they're all required for you now. But after 2022, you will need them because you want to go into medical. And in the first year, medical, in the first month itself, you will have to know most of the things. Don't worry, just now you don't need all these things. But a few things you should know. The heart is well known. And you know from the heart there begins dorsal aorta. And this is the dorsal aorta. You know, it starts from the left ventricle. And this dorsal aorta will pass between the two kidneys and will supply blood to the two kidneys. Then it will pass into the legs, and there are some arteries like femoral artery and sciatic artery. Femoral artery, you can easily remember because most of us know that this femur bone here, and therefore the artery that goes into that region is called as femoral artery, and there is a sciatic artery, and so on. Also there is a subclavian artery. Subclavian artery will go to the arm. It will have branches like brachial and radial. Some names I am trying to tell you. You don't have to worry. You don't have to remember this picture. You should have an idea. There is a very systematic supply of blood to all parts. Once again, the dorsal aorta, it passes between the two kidneys and it will provide blood supply to the kidneys. Then it will pass between the two kidneys and then it will travel. It will get bifurcated and the blood vessels will be supplied to the right up to the fingers of the foot and the toes and all others. And also there are some other organs in the body. There will be a branch that will go into the digestive system and all. I'm not going into those details. And there will be subclavian. The two arms will get the right and left subclavian arteries, and they will branch into various other smaller and larger branches, and ultimately the fingers. Everywhere, very beautiful supply. And from here there will be a connection established to the head, and that's called as carotid artery. Don't worry much to find out those levels. We have picked up this picture from Torah's book, and so it is easily available everywhere. Please don't try to remember everything. A few things here and there. Even if you don't remember the name of a single artery, still it's not going to matter immediately. There are arteries which are called as carotid arteries, and carotid arteries will be supplying branches to brain, eye, and some of the parts of the mouth. So the carotid supplies here, and then the subclavians here, and the femoral and the sciatic here. Those which are going to the kidney, that will be called as renal arteries, and heart itself will be supplied by coronary arteries. Don't get confused. These are carotids. Carotids are supplied to the brain, eyes, and the coronary arteries are being supplied to the heart. So although blood is passing through the heart is just as an ordinary organ like any other, and so it must have its own blood supply, that is coronary circulation. So this is the arterial system. Let's have a picture at the venous system now. Here is the venous system. Blood goes to all body parts. Remember, we are going to have some understanding of the capillaries little later on. We have a picture about the capillaries also, and that time you will understand that where the arteries end, there are capillaries. Arteries end into capillaries, and the capillaries reunite to make veins. Venous system doesn't start from heart; it ends at heart. Arterial system starts at heart, because arteries begin from heart and are distributed, the arteries. Veins don't start from heart. Veins begin from different body parts and end at the heart. So venous system ends at the heart because blood is being collected. Similar veins are there. Once again you will have femoral veins coming from the two limbs, and then you have renal veins which are coming from the two kidneys, and then you have these various veins, femoral with the subclavian veins and all, and then there will be jugular veins. These are called, instead of carotid vein arteries, now they are jugular veins which are collecting blood from the head and neck and all. And ultimately you have inferior vena cava, and this is superior vena cava. So blood from the limbs is collected, and then it forms the inferior vena cava, and the blood from the head region and neck region is collected, and they are called jugular veins, and the blood is collected here from various veins. They are variously known as brachial and several other things. Ultimately they form subclavian, and these subclavians on the two sides, they will join together, and ultimately you will have a superior vena cava, and they will all enter into the right atrium. So this is the typical venous system, venous circulation. So here we will now develop a concept of capillaries. Here this diagram shows this is the major artery. Artery is dividing into arterioles, and then ultimately the capillaries are there. What are capillaries? They are very, very fine, extremely minute branches through which blood will flow. What is the dimension? Capillaries can be as minute as 8 microns. I told you, even the red blood cells also is somewhere between 7 and 8 microns, and the capillaries are around that. And a beauty of this arrangement is that at a time only one red blood cell can pass through the capillary. For each capillary at a time only one RBC can pass. Naturally it will become very slow. When only one RBC is passing, all the RBCs are moving slowly. And what is the advantage of the slow movement? When the RBCs are moving slowly, oxygen can easily come out. And all these yellow things, orange things which are shown, they are the cells, tissue cells. And so they get all the oxygen which is being released from the blood that is flowing through the capillaries. It's not only the oxygen. These same capillaries have brought the different hormones, different vitamins, digested foods, and hundreds and thousands of things. Basically, one of the major functions of the blood is transport, and all the materials are transported to the tissues. So here at this finest level, the materials are coming out of the capillaries, and then the cells are producing several things. Cells are producing some excretory wastes, cells are all producing CO2 also, and that is entering here. So this is going slow. You know, this is a very slow motion of the blood. Arteries, the moving of the blood is fast. In capillaries it is slow. All these capillaries then unite. See the color difference. This is arterial blood, so arteries are giving out oxygen, and after that the oxygen is, carbon dioxide is added, so the blue color. But one thing, this can lead to misunderstanding. Some people may think that arterial blood is red and venous blood is blue. No, blood is always red, whether it is arterial or venous. Blood is red. This is a symbolic way to show. This is only a symbolic way to show. The only thing is that the venous blood can be probably slightly darker red, and arterial blood can be scarlet red, more brighter red. Otherwise, blood is always red. Don't think that this is blue blood. Actually, this is more symbolic. So that's how the blood enters into the small tributaries, and ultimately those tributaries they make the vein. These tributaries are called as venules. Artery divides into arterioles. Arterioles branch into capillaries. Capillaries reunite to form venules. Many venules join together to form veins, and you know the veins will run to the heart. So this is the capillary network. I think this is most important. Every organ in the body, whether it is brain or kidney, anywhere and everywhere, you must have a network of capillaries. These are many kilometers long in single body. We may probably talk about those numbers related to different systems of the body. Maybe that will provide you a list in the PDF form later on. But we have one more picture of the capillaries. Here it is. So this is how there is an artery. An artery will divide into arterioles. The arterioles will make a beautiful network of capillaries, and capillaries will join to form venules, and venules will then continue. And veins, you know, have this is a red color they have shown. Don't get confused. Don't think that how is it that this is red and this is also red. I told you it's all symbolic. So the vein, the blood is passing. This is valve. You know that veins have valves. Arteries don't have valves because blood is flowing fast. Veins, the blood is going towards heart, and there are difficulties because the flow of blood becomes slow. Because by the time the blood comes into the capillaries, the blood flow becomes very slow, and that slow blood has to go against gravity to the heart. So naturally, there are valves. So when the blood will be flowing through the veins, well, the blood tries to come back to because of the gravity force, but then the valves get closed. So overall, you can say arterial blood flow is fast and the venous blood flow is rather slow. That's about the capillaries. We have a pause. Interesting. We have learned about the arterial and venous system of humans, and here is the arterial system of frog. Frog, earthworm, cockroach, and humans. You have to learn these things to some extent, to some details. Look at the system very carefully. The three-chambered heart, and instead of one major pipe, there are two major pipes started. The systemic arches, they join together. This is the dorsal aorta which is passing through the kidneys. These are the femoral arteries. These are the subclavian arteries, and these are the carotid arteries. Oh my God! So whatever is there in human body is there in the frog's body also. I am sorry to say this. I should have said whatever is there in the frog body is there in the human body. Why I am trying to say this? Because evolution-wise, amphibia group is much older. So humans and mammals are comparatively recent. Only last 40-50 million years we have been here, mammals, and humans have been there hardly for the last 2-3 million years in this particular form. The amphibians are there for so many million years, and so this whatever system is there in amphibia, it got modified in the reptiles, and then it got modified in the mammals also. I want to say that there is a lot of homology. I have used the word once again. I think when we studied about certain other systems like human digestive system and this and that, I told you there is homology. What is homology? Homology is basic original similarity. For example, the arterial system of the frog and the arterial system of the human are homologous. Homologous because there is a basic similarity. Not only the arterial system, even the heart, frog's heart and human heart, mammalian heart are homologous. Some of you will say, no, it's not possible because frog's heart is 3-chambered and human heart is 4-chambered. Well, that is not a major difference. The basic design, the chambered heart, and that is homologous. In fact, it is considered that all the vertebrate hearts are homologous. You have seen one picture where the vertebrate hearts were shown, all vertebrate hearts, fish to mammals. It is all chambered design, and therefore it is all homologous. So heart is homologous, arterial system is homologous, venous system is homologous. I'll help you to compare the arterial system of the frog with the arterial system of the humans. Let us have some fun. Here it is. It will be very interesting to compare. So most of the designs later on, if you get time, you can study this in some details. So for every artery in the frog, there is an artery in the human. So if this is the dorsal aorta of the frog, there is a dorsal aorta here, you already know, and so on and so forth. If there is a femoral artery here, there is a femoral artery here, and so on. Very interesting. So I think we all vertebrate animals had some common ancestry, and therefore naturally we have common plan of the body design. I would like to project for you the venous system now. Here is the venous system of the frog. Some of the names you can read, and they are very similar to what we have. So femoral vein will be there, and this is the post cava, which is as good as inferior vena cava, and the jugular veins and the brachial veins, which make the subscapular innominate together, join together to form the precava. Basic design is all the same. And let us once again have this compared with the venous system of the humans. And here I have the venous system of the humans. I'm just trying to put them together, and because now we have some limitations of time, I may not be able to comment more on these things, but if by chance if you are able to go through these things in details at now or later on, you'll find that almost every vein here is represented in the human body. It's not only between frog and human. All vertebrates will have common plan of arterial and venous system, and some basic plan of the heart will also be same. This is quite good, and I think I can show you now earthworm system. Earthworm circulatory system is equally complicated. Oh my God, you never expected that earthworm system will be so complicated. Earthworms we can easily think of kind of worms which are seen many times during the rainy season, and we don't ever bother them. My God, this is only in the first 13 segments. Earthworm can have more than 100 segments, and the system of the blood vessels behind the 13 segments has a different plan. But even in the first 13 segments, there is so much. This is going to be there in your books later on in the 11th, and details to some extent you are going to study. Looking at all these things, you may be feeling, oh my God, biology is going to be tougher. No, it's not tougher. As long as you are with us at Learning Center, you don't have to bother. We'll make sure that everything will be palatable to you, digestible to you. Conditions apply. Rather conditions, you must be attentive, you must be sincere, you must be honest, and you must be regular in your studies. Then everything becomes easy. Yeah, but yes, it is true that you just cannot take biology for granted. You'll have to spend good amount of time for that. You don't have to remember everything, and these days you are not expected to draw everything, but this is something that you are going to study in 11. So there are lots of blood vessels, and yeah, we will show the dissections of those things at a later stage, and that time I think you can understand better. The point is, a simple Annelida class, Annelida phylum, in which the earthworm, to which the earthworm belongs, even that one has got complicated blood vessels. One thing, such a system, whether it is earthworm, whether it is frog, or whether it is humans, the blood is flowing continuously through blood vessels. It's not coming out, because we know that arteries end into capillaries, and capillaries end into veins, and veins join heart. So blood is not coming out anywhere. This kind of circulation is popularly being called as closed circulatory system. I will be commenting on closed circulatory system and open system. I'm just trying to say that the frogs, the humans, the birds, the reptiles, fishes, and even the earthworm. They have blood flowing through tubes, and therefore it's called as closed circulatory system. I think before we stop talking about the blood vessels, can we have a picture about the lymphatic system? I think we can have a picture about lymphatic system. Actually, I wanted to show you the lymphatic system, but I think we can look at it after a while, because in continuation of what I was earlier trying to say, this is the closed system and this is the open system. Circulatory system in animals is of two types: a closed system and an open system. A closed system is that which I just now explained in humans, in earthworm and in frog, that blood is coming from heart, and in case of vertebrates, the arteries are divided into arterioles, and then they are divided into capillaries, and cells are very close to the capillaries, and then all the deoxygenated blood is being collected, and all that is going back to the heart. So this is arterial system, which is ending into capillaries, and from capillaries venous system begins, and that goes back to the blood goes back to the heart. So arterial system starts from heart and venous system begins from capillaries, and no drop of blood is likely to come out. This is what is called as closed system. And because all vertebrates have closed system, we have closed system, it is obvious for us to think that yes, this is the best system, because cells are getting a very close supply of the blood from the capillaries. Fine. But remember, there is an open system which is probably more common in the animals. You will be surprised. This is the open system which is present in arthropoda and to some extent in mollusca. My God, arthropoda and mollusca. You know, arthropoda is the biggest phylum. Out of so many animals, 75% animals belong to arthropoda. That means majority of the animals in this world have open system. Add to that the second biggest phylum is mollusca, and many mollusks also have this system, open system. What do you mean by open system? There is a tubular heart, and tubular heart is not chambered. You know, heart is also two types. Chambered heart in vertebrates, 2, 3 chambers, 4 chambers. Tubular heart is series of pouches one behind another, and that is on the dorsal side. That is usually the tubular heart generally. And the heart is like a tube, fine. But the blood from the tubular heart, it enters into body cavities, and the body cavities have organs, and organs have cells. My God, there are no capillaries here. The major difference in the closed and open circulatory system is here there are capillaries in the closed system, and in the open system there are no capillaries. Blood is flowing, and the blood gets collected, and it goes back to the tubular heart. So from tubular heart, the blood is poured around the different organs where the tissue cells are there, and then it is collected. We never think of these things, but let me tell you, majority of the animals in the world, all arthropods and majority of the mollusca have open system. If you compare these two, because we have this system, you are comfortable with this understanding. But let me tell you, here the cells have more close contact with the blood. Okay, the blood is not red. Here the blood is usually red, mostly red of course, because closed system. Even earthworm it is red, but here it is not red. That doesn't matter. But there is no obstacle. You know, the cells and the blood, the cells are directly in contact with blood. So exchange is better. Exchange of material between blood and cells is better here. It seems that open system is equally good, probably more good in the animal world. So that's about the two types of systems. I think we will now try to pick up a different thing, a little bit of revision of few things, and we'll go back to this screen again. I think we have just learnt about the open and closed system, and on this board we have already learned the cardiac cycle and double circulation. I think this can be neglected. Come back to this diagram once again. You can see here the blood is coming from heart in our own case, or in case of vertebrates, and arteries dividing into arterioles, and arterioles are divided into very fine capillaries, and the capillaries are the arterial part and the venous part of the capillaries. Capillaries are reuniting to form venules, and this is the beginning of the venous system, and venules join together to form veins, and the veins they go back to heart, and blood is all flowing through the closed system. Tissue cells are there, of course, and there is lymph. Now let me talk about the lymph. Lymph is something which is colorless. I think I had mentioned it earlier, and lymph is derived from blood. See, many materials are coming out of the blood capillaries, and many materials are coming out of the body cells. So body cells and blood, they are having lots of exchanges. Not everything goes back into blood. Certain things which are escaping from blood and escaping from cells, depending on the need of the local tissues, those things will enter into another system known as lymphatic system. This is a lymph capillary. This yellow thing is a lymph capillary. So, where the blood capillaries are there and cells are there, certain materials from the blood and the cells will enter into lymph. So, you can say lymph is derived mainly from blood. It is colorless. It has got lymphocytes, but it doesn't have red blood cells, and it moves slowly. This is a lymph capillary which is closed at one end. And now these lymph capillaries, many of them are again very minute. They will unite together and they will form a lymphatic system, and we will now have a look at the lymphatic system from the screen. So here is the lymphatic system. Lymphatic system consists of, once again, tubes, the lymph vessels. As you can see, from all body parts, lymph vessels are there, and just now we have seen that there are fine lymph capillaries. Those lymph capillaries collect the material in the form of lymph from the tissues near the cells, and from near the blood capillaries, materials are entering into the lymph capillaries. Lymph capillaries unite together to form bigger tubes, lymph vessels, and all these lymph vessels ultimately bring the lymph and pour it back to the venous blood. The lymph is formed from blood and it goes back to blood. This is continuously going on. Then you may say, what is the need of the lymphatic system? Well, lymphatic system has double purpose. One purpose is that there are certain substances which are preferably carried by the lymph. I'll give you one example. Think of the lipid digestion products. You know already that fatty acids and glycerol, they enter into the lymphatics. It's convenient to carry those things from the lymph system. Like that, there are many things in the body which are better carried through the lymphatic system. Second major purpose is, as you can see, this lymphatic system has many swellings here, everywhere. And this is one of the major swelling. This is a very important organ known as spleen. This spleen is one of the major swelling, and these are smaller swellings. At least 200 such swellings are there, and those are called as lymph nodes. So on the way back to the blood, the lymph passes through lymphatic nodes. Very important things. Lymphatic nodes are the places where lymphocytes are being produced, and lymphocytes are important because the lymphocytes, I think you must be remembering, I told you lymphocytes are of two types, B and T types, and lymphocytes make a lot of difference. They provide us best immune power. There is a thymus gland also, as you can see here. You must have heard of thymus. Don't get confused. Thyroid is different, which is here. It is not shown here. This is thymus gland. Just like spleen is a lymphoid organ, thymus is also a lymphoid organ, and it is the thymus which has got one special role in producing special defense cells known as T lymphocyte, because it is producing thymus, it is called as T lymphocytes. So lots of things are there. It means that while the lymph is flowing back to the blood, lot of lymphocytes are being added to the blood. Moreover, a lot of antibodies are being produced. I mean, without lymphatic system, it may not be possible for us to have the kind of immune response, the defense response that can show to the environment, to the germs. So that's about the lymphatic system. This is how it is explained here, that only this much part is drained by what is called as right lymphatic duct, and the green part all it is drained by what is called as the thoracic duct. You don't need those details, but just like you know, there is a superior and inferior vena cava for collecting the venous blood. Similarly, there are systems to collect the lymph. You don't worry much about this. So this is what is called the lymphatic system. It seems it's again going to be a long session, the circulatory systems and circulation. There's a lot of complexity in the living world, and one of the reasons of complexity is variety. You can see a lot of variety. Everything seems to be designed as per need and so on. I'll quickly say what we have learned till now. We started with blood, and then we took into account the details of the heart and its working. Then I think we went for heart of the frog and evolution of the heart, fine. And then we considered arterial and venous system, and then the open and closed system, and we are all doing more details of the closed system, and we have considered just now lymphatic system also. I think now the session comes to an end in the next 10 minutes or so, but I would like to have a few more comments done for the blood, because I think there will be some better pictures available for us with the help of this overhead projector. So here you can now look at the red blood cells. They appear to be disc-like. 7.2, you can say anywhere 7.2 or 7 to 8 microns, that is their diameter, and they are concave. So there is no nucleus. This is not nucleus, they are concave. So the inner circle doesn't represent nucleus, the concavity. So they are biconcave, both sides they are having a cavity, as you can see here. And the red blood cells are very well known to you. There are millions of hemoglobin molecules, and they carry oxygen from lungs to tissues, and they carry carbon dioxide. I mean, the carbon dioxide transport is interesting, and there are many details, and hemoglobin is required even for carbon dioxide transport, and that's how the red blood cells. We can have a very quick look at the white blood cells, but to be very frank, they are not white in color, and the picture that I am going to show you are mostly the white blood cells. Also in the beginning there is a erythrocyte, the red blood cell. I think we are talking about it. Think of all these white blood cells. They are not white, they are leukocytes, colorless. Neutrophil. This is also a neutrophil. Two designs of the neutrophil. There are more designs. Eosinophil. Basophil. Two types of lymphocytes, small lymphocyte, large lymphocyte, and monocyte. Would you like to repeat after me? I'll say and you repeat. Neutrophil. Again, one more type of neutrophil. Eosinophil. Say it all.
Eosinophil.
Basophil.
Basophil.
Small lymphocyte and large lymphocyte.
Small lymphocyte and large lymphocyte.
And this is monocyte. One of the biggest is the monocyte. There are typical shapes of the nuclei, and from that it is possible to identify. I think we are going to have one more slide on the main screen where we are going to see some of the cells, colored cells later on. So, you know, these are all granulocytes because they have granules, as you can see. And here these are all granulocytes because their cytoplasm is clear. They don't have granules. They appear to be all circular here, but actually each one of them is irregular. When you take them on a slide, and it is possible to make a slide, we will try to show you sometime later on how to make a slide of the blood. But when they are taken on the slide, they appear to be spherical actually, so they appear to be circular. Basically, they become spherical. But in reality, when they are moving in the blood, they are amoeba-like, so they are irregular in shape. I think I'll show you one slide here where you can think of the way the WBCs, they are able to come out of the blood capillaries. I think this was told to you when I was trying to explain the white blood cells. A white blood cell has mechanism to come out of the blood capillary. Why? Because germs, wherever they are, they will go and kill them. So they suppose a neutrophil. This appears to be a neutrophil because the multi-shaped multi-lobe nucleus is there. Suppose there is a germ here. There is a proper sense available, chemical sense, because of which the neutrophil will be attracted to the germ and it will eat the germ. So this is what is called as diapedisis. Don't worry about the terms. It is the ability of the white blood cells to come out. Red blood cells cannot do it, but white blood cells can come out. White blood cells can also move in the direction opposite to the direction of the blood, and so they are very different. If you are trying to make two columns, and yes, I can see some of you are interested in making two columns to write the difference between RBC and WBC. So if that difference is to be done by us, I think I can point out a few things together. If you are making two columns, just now you are making good, because I think many of you are sitting with the notebook. I will constantly remind you that foundation course will be successful only with your cooperation, only when you are serious and sincere about it, only when you sit in front of your screen with a notebook, and it will have much better experience than the regular classroom, to be very frank, because here is one teacher and one student. I am there and you are there, no one else is there, right? Okay, so make two columns, RBC, WBC. What are the points then? Red blood cells are red, white blood cells are white. No, don't write that, because white blood cells are colorless. Red blood cells are circular biconcave, white blood cells are amoeboid. Red blood cells are without nucleus, at least in mammals, and white blood cells have nucleus, and the nucleus has different shapes. Red blood cells are much more in number. Millions per cubic millimeter, there are millions of red blood cells. But white blood cells are only in thousands per cubic millimeter. So one is in millions, one is in thousands per cubic millimeter, I am saying. Life of a red blood cell may be 100 days plus minus. And in life of a WBC, well, WBCs are soldiers. And why you are asking of the life of a soldier? It can be 1 day, it can be 10 days, depends. They are continuously at war, and usually they will have a much shorter lifespan than the RBCs. Both RBCs and WBCs, and also the platelets, the small things, they are all being produced in the bone marrow. So differences, the comparisons are important way to learn, that provides us an opportunity to be more analytical, because analysis is a very important part of science. I think we will take a pause here. So I think we will have few more pictures, colored pictures about the blood cells. Okay, let's go ahead. Basically, you are aware that 45%, here they are showing 41 because they are also showing platelets here, and so this is all taken together. 45% are blood cells and 55% is all the plasma, and it is here in the component of the cells that you have RBCs, WBCs and platelets. Let's have a look at that. Look at this one. This is eosinophil, actual preparation, and the cells will look like this after staining. The basophil and the neutrophil. The three together they have granules. Out of these, neutrophils are very important because it is these cells which are phagocytic. What do you mean by that? Phagocytic means they are able to attack the germs directly. Then you have lymphocytes. This is a small lymphocyte shown, and large lymphocyte they have not shown. It's okay, but lymphocytes are those which are of two types, B and T lymphocytes. The nucleus is so big. And monocytes are also scavengers. They are just like neutrophils. They directly attack the germs. There are some special roles of the eosinophil basophil, but I think it will be out of place to mention all those here. You have once again the blood smear. All these are red blood cells, and some of these here you can observe these are the leukocytes. Small things are all RBCs, and these bigger ones are the lymphocytes, neutrophils and others. And these are the bigger pictures of the same thing. You can see the neutrophil here, and you can see a big cell, the monocyte here, and this is a B lymphocyte and this is a T lymphocyte. And I think you have just one more picture to remember better the blood cells. With this, I think now we are going to proceed with, oh my god, okay, we have hemoglobin, RBC and hemoglobin, that's nice. There are millions, I've been telling that millions of hemoglobin molecule in a given red blood cell. This is a red blood cell which is biconcave, and inside it there are millions of hemoglobin molecule. Hemoglobin is a protein, and it is a protein that contains iron. This is a hemoglobin molecule. You know, it's a protein, so it has got chains. I think you are generally aware that there are amino acids making a chain, and this is a polypeptide chain 1, 2, 3 and 4. 4 chains of amino acids known as polypeptide chains make one hemoglobin molecule, and one hemoglobin molecule has 4 units of iron. It will look like this, and that's how it is, because of the iron that the hemoglobin is able to carry the oxygen. So this is RBC and hemoglobin molecule. There are so many details, but I think we have limitations of time and purpose also. Oh my god, it's very, very interesting, a little bit of tiring, but more interesting than tiring. I'm saying tiring because it is taking a long time to present before you, say 1.5 hour lecture of this kind, where I have to move from one place to another and take shots at different places. Well, it's time taking. It takes at least 5 to 6 hours to record a lecture of 1.5 hour. Well, we are a team of 3, and myself trying to take the lecture, and you have Rishikesh sir and the Kaustubh sir here, and of course I told you they will also be coming with me here on the stage in the subsequent days. But it is taking time. Moreover, this is something totally new to us. We never, I told you, in the last 47 years, I had never thought of doing these things. But then it's interesting. It is taking time. We have to get ready with so many things. We have to make sure that everything is in place. And that takes another few hours. And actual recording takes 5 to 6 hours. And it is not only for you. At the same time, we are doing it for the senior batch. Your senior batch who are already in 12th. Their course started in January, and suddenly from March 20 it was declared that is a lockdown, and then we are working for them. Their class is also regularly going on in the same way, actual teaching that is going on just now here for you in the same way the 12th class students, and I think for them also we'll have to pull it, I don't know till when, maybe even September, October, whatever, and by that time, because it is 12, the portions will get over, we'll have to get over. So I think every obstacle is an opportunity. So we are taking it up. So we are working every week for you. We are working every week for your senior batch who are already having the regular lessons going on. They are in 12 now. And of course, we are also working for your super seniors, those who are going to appear for NEET this year. So we are enjoying the work, no doubt, but please help us, because it is possible that many times we may not be able to stick to the timetable that we have given. We're trying our level best, but most of the time, if we fail to deliver the recording on that particular day, you will please excuse us. So keep on checking your mails. We are giving you messages also. Overall, I am sure you are enjoying. We have been in touch with many students and many parents, and it seems that they say that this kind of method that is being used is more useful to them. I don't know what is your experience. You can compare your experience of this method with other methods which is probably used by other subjects. Maybe that there are some advantages of the Google Meet and Zoom and all, but I think it is possible for a small group, whereas ours is a very big group. At a time, more than 200 students are listening to all this, and interactions are not possible that way. Yeah, the only shortage of this particular method of online is a direct one-to-one doubt solving may not be possible. But let me tell you, there was a mail given to you, and it was clearly written that it is possible that we can start getting your queries through the same platform. Use the Google platform for Google Classroom platform for writing your doubts, and we will put them together, and one session I can hold only for clearing the doubts, and some other ways also we can do it. Okay, so whatever is the shortage, that's the only small shortage which we can make up, and this becomes a foolproof system. Okay, so please cooperate. I think now I'm just going to say something very special about some of the special animals, the porifera and echinodermata. A porifera, you know, sponges, and that's quite a lower group, and you are not expecting any heart in them and no blood and no blood vessels, but there is something which helps their life, and it is what is called as canal system. These sponges have what are called as canal systems. You know, probably you have read in the lesson of animal classification that the sponges have small apertures on their body, the pores, and through the pore water enters. There are different designs of the sponges. Ultimately, water enters into their bodies. Some sponges are like this, some are like this, some are more complicated like this. All these are canal system. What do you mean by canal system? Canal system is a pathway by which the water enters into the sponge body and leaves. Small apertures are there in the body, water enters, and from a bigger aperture known as osculum, the water is going out. And along with the incoming water, oxygen is brought. Along with the incoming water, food particles are brought, and the cells will use the oxygen, release the carbon dioxide, use the food. That's it. So simple life. Anyway, they don't have even tissues. Forget organs and systems. So that's one thing. So lots of options, lot of varieties available. With that, now we can go for one more diagram of the system, I mean the canal system, and then we shall go for echinodermata. Firstly, let us have once again canal system in a sponge. I told you already, through small pores the water will enter, and through the big hole it will go out. Don't worry about these labels, you know, because these are all diagrams adapted from the different sources. We can't avoid labels. Those labels are not required for it, don't worry. We will now go for the very special, very special system in echinodermata. You know, echinodermata are really very special. They have radial symmetry. Let's look at water vascular system in a starfish. A starfish everybody knows. And starfish inside have a system which fulfills so many purposes. This is water vascular system. There is no blood, nothing. So the ocean water enters from here, and from here it goes into a ring-like structure, ring canal. Ocean water enters here, then it goes into this ring-like structure, the ring canal. From there it will go into radial canals, and then from there it will go into what are called as the tube feet. Some of you may be knowing, some of you may not be knowing the characters of Echinodermata. We are going to learn the animal classification also in details in the regular course, and that time we are going to comment on this system again. So the water passes through all this system and it comes out to the tube feet. And what are the advantages? Well, respiration. Along with the water, carbon dioxide and oxygen are exchanged, and along with the incoming water, well, there are many things, unwanted substances can also be given out, and moreover, movement, locomotion. Pressures are generated by the tube feet, which are very specially designed, and then the organism is able to move. So this is water vascular system. Actually, Echinodermata is a higher group as compared to Porifera. So I don't want you to compare Porifera with Echinodermata. This system is quite complicated. This is a very, very special case, and that's how I think water vascular system. One more picture you can have and look at the water vascular system of the Porifera. Same thing. Water is entering here, and I think this is a okay. I just have a look at the canals. These are the canals here, and the tube feet. I think that nothing very special. That's it. So, and then we will gather some information about blood groups. A very popular topic, and some of you must be knowing a few points about blood groups. But let me tell you, it is also equally possible that there are lots of confusions and misunderstandings about blood group. What are blood groups? Basically, blood groups are nothing but differences. You can say these are genetic differences at chemical level. We are all aware that people have different blood groups. Some have A, some have B, some have AB, and some have O. Amongst yourself also, each one of you will have either A or B or AB or O, and these blood groups are because they are kind of inherited. Of course, it's not that simple because both parents have same blood group or different blood groups, and still your blood group may be different. For example, one parent may have the blood group A, another may have the blood group B, and you may have the AB, but it is possible that you may have O also because depends whether it is homozygous A or heterozygous A. I think we are not going into the genetic details, but I just want to give you a hint here that blood groups are hereditary, so they are genetic, and there are chemical differences. These are due to certain chemicals. I'll show this here. Here is a red blood cell, and on the surface of red blood cell, there are antigens for the blood group. What do you mean by antigen? Antigens, well, they are again chemicals. If you know the word glycoprotein, then you can understand. If you don't understand glycoprotein, okay, you think that it's some kind of protein, or you can say some kind of chemical. Actually, there are two types of chemicals, A chemical and B chemical. So antigens may be A or B type. Two chemicals. Some people on their red blood cell surface have only A type of antigen. Some people on their red blood cell surface they have only B type of antigen. And some people will have both mixture of A and B. Once again, some people have only A type of antigen, Some people will have only B type of antigen, and some people will have a mixture of A plus B. And there will be many people who have red blood cells without any antigen. They have neither A nor B. Now think, according to antigen, the blood groups are decided. I can make a chart here. Antigen and the blood group. If the antigen is A, blood group is A. Simple to remember. If antigen which is present on the red blood cell is B, the blood group is B. Very simple. And what are A and B? They are chemicals. They are called as antigens. Different glycoproteins, slightly different chemically. If antigens are A and B together, then the blood group becomes AB. And if there is nothing, nil, no antigens, then the blood group becomes 0. This is A, this is B, this is AB, this is 0. But 0 looks like O. That's why the blood group is called as O. Actually it is 0. Why 0? Antigen is absent. So instead of 0, people call it as O. So this is how you have 4 blood groups. A, B, AB and O. That's so simple. Along with antigens, there are antibodies also. And if I write them, you will find it slightly confusing. So I want to avoid it. No, I will show it. Some of you can record it. Antibody. I'll write a column here, antibody. Along with the antigen, each person has a corresponding antibody. And yes, here I think you will feel confused, but in the long run you are going to learn it. Those people who have blood group A, they will have antigen A and antibody B. Oh my God, this is too much. Those people who have blood group B, on the red blood cell phase, they have antigen B and antibody A opposite. There is a purpose behind this. Those people who have blood group AB, they have a mixture of both antigens A and B. And can you imagine which will be the antibodies? Nil. No antibodies. And those people who have blood group O, they have no antigens. Antigens absent. So yes, you are sure, you are correct. They will have both antibodies A and B. Oh my God. So what is blood group A? Antigen A, antibody B. What is blood group B? Antigen B, antibody A. What is blood group AB? Both antigens present but no corresponding antibody. And what is blood group O? No antigen but both antibodies. A lot of chemical differences generated. This is called as variation, physiological variation. Now it is very difficult to say why at all, what is the use? No simple answer for this. What is the use that somebody has A and somebody has B? And for example, mine is O and somebody else you are listening, some of you may be AB. What is the use? Why to have a particular antigen and antibody? Well, I can provide you an example. Why some people have a straight nose and some people have a nose which is odd like mine. Why some people have earlobes attached and many people have earlobes free. You know, there are so many differences in the facial features, and some people have curly hair and some people have straight hair. Some people have darker skin, some have a fairer skin. Why all this? This variation. Please understand, a very important feature of life is variation. Variation may be in morphology, it can be in anatomy, and it can be physiology. This is a physiological variation. You cannot ask a question, what is the use? Well, it seems that nature creates variations. You know, when you look at the plant world, the leaves have so many different shapes. What is the use? Well, that particular shape seems to be good there. Within the human population, if we have these kinds of physiological differences, it's okay. Why only this? At cell level also a lot of physiological differences are there. But then I cannot stop the story of blood group here, because you want to know something about positive and negative. So the blood groups may be A, B, AB, O, and also positive and negative. Here is a lot of confusion. I just put up here, there is what is called as RH system of blood groups. This is ABO system. And here is RH system of blood groups. What do you mean by RH? Well, many of you have heard the story. This RH stands for name of a monkey by the name Rhesus. Why at all monkey comes up here? Well, and this particular second series of blood groups was first discovered in monkeys, and then it appeared that the same system is appearing present in our human body. Rhesus is due to D antigen. If D antigen is present, then it is RH positive. What is D antigen? Well, just like A and B antigen, there is a D antigen. And then what will be negative? If the D antigen is absent, then it will be called as RH negative. Simple. You have two systems. One is ABO system, one is RH system. Two systems of blood groups. Both are existing at a time in our body. For example, really speaking, I am O positive. It means I have no antigens. I am O positive, so I don't have any antigens on my red cells. A and B antigens are absent from the ABO system, but I have D antigen present. So what will be the nature of my red blood cell? On my red cells, there will be only D antigen. A and B are absent. So my blood group becomes O positive. Somebody, those of you who are listening, suppose somebody's blood group is A positive. What does it mean? You have A antigen plus D antigen. So if you have A and D, you become A positive. If somebody is A negative, what does it mean? That you have A negative means what? You have A antigen. So you are having A blood group, but there is no D antigen, so it becomes A negative. And like this. Then what is AB positive? How many antigens will be present? Just think. A, B blood group will have A and B antigens and D antigen present. What is AB negative? AB negative, that means A and B both antigens present. That's how the blood group is AB. But D antigen is absent. You find it little complicated? It is little complicated. Don't worry much about it. But yes, this is going to be there for you in more than these details. Some of you already know it and some of you don't know it. It's okay, fine. But so, for a blood group, this is a chemical difference. These are kind of variations, and two blood group systems exist. And let me tell you, blood groups are present in other animals also. It's not only in humans. They are present in other animals also. And in the humans also, it is not only ABO and RS system. There are many other blood group systems also. But from practical point of view, from medical point of view, I think only these two systems are considered seriously. And there are many smaller blood groups like Bombay blood group and all that. We are not going to consider everything. Coming lastly about the donation. And you are all aware that a person with blood group A can donate blood to A B person can donate it to B, AB can donate it to AB, O can donate it to O. That is most safe. A to A, B to B. AB to AB, O to O. However, A type of blood is okay with A along with AB. B type of blood is okay with B along with AB. It means AB can receive from A, AB can receive from B. AB's blood, the blood a person, a donor who is AB, the blood is okay only for AB. That means the person with blood group AB can receive from O, can receive the blood of B type, can of course receive the blood of AB type. How about the O blood group? My blood group? Well, I can be a donor for O. I can also donate my blood to A, to B and to AB. Interesting. So that is how O is called as universal donor. And AB is called as universal receiver because AB can receive from A, AB can receive from B and AB can receive from AB as well as O. So AB has lots of options. So those of you who have AB blood group, you can receive blood from all. And those who have O blood group, you can donate it to all. But remember, person with O blood group can receive only from O, not from A, not from B, not from AB. So a person with O blood group can receive blood from O, but can donate to all. So it's very popular that AB is universal receiver and O is universal donor. About the blood group, there can be many other details, but then the donation is something which is being done, which is expected to be done by a lot of people. The more and more blood is donated, there are blood banks and it can be stored, and there are certain other issues related to the blood groups. But no confusions, please. Please understand, it is a chemical difference. It's normal to have different blood groups. Blood groups can be tested with some chemicals. These are called as blood sera, and this is anti-A material, anti-A serum. This is anti-B serum, and this is anti-D serum. So we are going to use this blood sera for blood group testing, and I think you can expect this today evening. This portion is being released to you for your studies by tomorrow, maybe 12 or 1 o'clock. I think in the evening you can have a demonstration. We'll arrange a small demonstration that we can show you the blood group tasting and the heart of the goat and a little thing here and there. So some demonstration will be there. It means that according to timetable we were supposed to do excretory system tomorrow, right? But I think it's not possible. We don't want to rush through the things. We want that everything should be nicely understood by you people, and you must consider that the details which are being presented to you are going to be extremely helpful later also. So there is no hurry. There is a lot of time available with us. So don't worry. Actually, NEET preparation has started. If parents are listening, it's good. If not, tell them that NEET preparation has begun. They will feel nice. Why? Because they are all worried about your education. They are all worried that this lockdown and this current epidemic problem, is it going to have a negative effect? No, not at all. In fact, it will have better effect. This is the way you are going to learn, and many things we are going to demonstrate side by side. Absolutely no problem. Okay? So that's it. The worksheets are already provided to you. Please give us your feedbacks in what way you like this or dislike this kind of work, and also yes, write to us your doubts, and we will arrange a special session when all your doubts of the earlier sessions can also be discussed. I think in spite of the odd situation world over now, still we are enjoying education, and being involved in education, teachers and students, is an important thing. It's a national service. Okay, so take care of yourself.